on the screen. It's in the PowerPoint. I don't believe it's in your lecture notes, but it kind of ties the LH and FSH changes and the estrogen and progesterone levels and the follicular and uterine components, which is essentially what today's lecture so far has covered. All right, so that I have each of these images separately in your lecture notes, but this puts it all together on one page. So now, today we're going to cover the process, finish up with the process of the oocyte moving into the oviduct and what happens as it meets up with the sperm. Okay? So ovulation has occurred. This is where we stopped right here, just before break. And this would be the corpus hemorrhagicum. The mature follicle has ruptured. And at this, just prior to ovulation, the fimbriae move close to the ovary, increasing the probability, and they're modal, they're moving, increasing the probability that the the oocyte, secondary oocyte and its surrounding cells will be taken up into the oviduct. So when this occurs, now this is going to move through the oviduct. And remember I said, frozen on me. Fertilization typically occurs within the ampulla, right? If it occurs too early, then the developing cells, multiplying cells, will be ready for implantation before they actually reach the uterine cavity. And if fertilization occurs too close to the uterus, then by the time the developing and multiplying cells have reached a stage where they can settle into the stratum functionalis, they're already on their way out the cervical canal. So we'll come back and look at that again tomorrow when we talk about implantation. Sometimes implantation takes place down near the internal os, and as the placenta develops, it actually crosses over partially or completely the internal os, and that makes the mother at risk for hemorrhage because it's not attached to a secure stratum functionalis there, and especially as the fetus grows larger, there can be bleeding and separation prior of the placenta from the stratum functionalis tissue prior to the baby being ready to be born. Okay, it's known as placenta previa. And so normally the implantation is on the posterior wall of the uterine cavity, about a third of the way down from the top of the fundus. And so it probably had fertilized a little bit too late and was closer down to the opening of the internal os when implantation actually took place. But we'll look at that tomorrow. So as the sperm is moving towards the egg, remember I said they pool just outside the entrance to the oviduct at the isthmus, at that end of the oviduct. And about a group of about 100,000 will move through, swimming and being propelled by peristalsis. And the opposite direction is the egg is moving. And the sperm have to undergo a process. Two different things occur. As you can see this at the top of page 127, capacitation and the acrosome reaction. So capacitation, this is beyond what occurs in the epididymis. So we talked about the sperm maturing at the tail of the epididymis. This process involves enzymes becoming activated or able to be activated, and some loss of membrane stability. By that, they're more likely to fuse with other membranes. So that process occurs within the epididymis. Okay? However, capacitation occurs within the female reproductive tract, and that was one of the critical structures that was difficult for reproductive scientists to accomplish in vitro fertilization. Because you can't just put the sperm in an egg and saline solution and expect fertilization to occur. Primarily the problem was with the sperm. All right? They hadn't undergone this process of capacitation. And so the lab that I was volunteering in over at Davis was one of the key labs in identifying progesterone levels as one of the elements required for changes in the sperm that allow it to fertilize the egg. So there's proper pH, there's other enzymes and hormones, but progesterone was one of the last ones that was had to be added. So there wasn't sufficient just in putting the egg in the dish. The milieu, the fluid of the in vitro dish had to have sufficient progesterone. Okay? So that causes changes in the sperm and loss of some of the membrane proteins that are stabilizing that outer plasma membrane and allows it to undergo the acrosome reaction. So capacitation are essentially chemical changes, structural changes because of chemical changes that prepare the sperm for two events, acrosome reaction and then binding to the egg membrane itself, the plasma membrane of the egg. So if you recall, when we looked at the sperm structure, we had the head, plasma membrane, we had the nucleus, and then we had the acrosome or acrosomal sac, which lay over that and had the digestive enzymes required for getting through that zona pellucida. Okay. So what occurs in the acrosome reaction, and some of these cells will undergo the acrosome reaction before they actually get to the egg and release their enzymes that allows the sperm to form essentially digestive tunnels to pass through the corona radiata and all the follicular cells around it. All right, some of the cells will not undergo their acrosomal reaction until they actually get to the zona pellucida. So that's why it requires so many of the sperm to do so. So the acrosomal reaction event results in the destabilization of the membranes, and we have the plasma membrane of the sperm, the outer cell. We have the outer acrosomal reaction, acrosomal membrane. And we have the inner acrosomal membrane. And in this destabilization, that allows these membranes to separate. And they don't just separate into little globs, they actually form vesicles. Because remember, they have lipid layers that don't like water, so they kind of join together. So this would close off, and we would end up with little sacs forming as this membrane disintegrates, and there's nothing holding them in place anymore. All right, so as these little sacs are formed between the plasma membrane and the acrosomal membrane, they actually start to wash away. And so we have this little stub here formed by the fusion of the outer acrosomal membrane and the plasma membrane. And we have the same thing up here. And so notice that the acrosome is now open and exposed. So all of the acrosomal enzymes are now free to interact with the surrounding granulosa cells or the zona pellucida. So there used to be, I had a connection to it via a link on my slide, but software has changed. It's not working anymore, so let's see if I can. It may have disappeared. Bless you.
Sorry.
Go. So what you're seeing in that kind of white line are those vesicles forming and then getting washed away from the surface. So this thickened region right here will be the fusion of the plasma membrane and the outer acrosomal membrane occurs. And so you can see then the black surface that remains. So here we're seeing the plasma membrane, that it's fusing with the outer acrosomal membrane, and this dark surface that remains is not the membrane of the nucleus, but the inner acrosomal membrane. And so these little sacs are forming and being flushed away, and there you'll see an increase in the free surface of the inner acrosomal membrane. That inner acrosomal membrane is what actually fuses with the egg. So that's where the membrane and egg fusion takes place. So this type of information, what happens with the chromosomal material, we'll talk about tomorrow. But here we have the sperm coming in, breaking down, many of them just releasing their acrosomal enzymes to break down the follicular cells and the corona radiata. One of them making contact with the zona pellucida, several of them, each of them trying to digest. So it's not like we need 20 million to digest through the zona pellucida. Each sperm has to digest its own way. Those extra cells are making their way through the corona radiata. So these are on a race, they don't realize it, to make their way through the zona pellucida and get to the egg. Now some of them will get to the first or second polar body, all right? But even if they do, it doesn't have enough cytoplasm to support any significant mitosis, and so no additional organism is going to develop. So same thing, this is actually shown on a sea urchin, all right? So here comes in the sperm that's actually going to penetrate through the zona pellucida. So it comes in through here. When its membrane contacts it, the zona pellucida protein is identified as a ZP for zona pellucida, a ZP3, and that triggers the acrosomal reaction of the sperm that's going to make its way through. So here you see the little stubby components. It digests its way through that zona pellucida, and in human sperm anyway, comes in sideways and binds to the egg along this surface. Okay, so it doesn't jam itself right in like a rocket missile or something like that. It comes in sideways, and the inner acrosomal membrane will bind to integrin. This is on your lecture notes. Integrin alpha 6 beta 1 protein on page 128. So when that occurs, two events result called that result in a block to polyspermy. Polyspermy would result with too much chromosomal material in the egg, and instead of now having 2N, we would end up with 3N or 4N or however many multiple sperm added their genetic material, and that would not be capable, that would not be compatible with life. So there's a fast block to polyspermy, and then there's a slower block. Okay? And you'll see some of these events in the sea urchin. So what happens, what seems to be actually species specific is that ZP3 protein. So one sperm from a different species won't interact with the ZP3 protein. But once it gets through the zona pellucida, it doesn't seem to care. Whether it's a horse egg or a cow egg or whatever. So the species-specific protection is in that zona pellucida. But it makes its way through, binds to the integrin alpha-6 beta-1, and one of the things that occurs are sodium channels open. Remember, sodium is higher in concentration outside the cell than inside the cell. And so we see a spike, an elevation in the positive number of charges inside the cell, the positive charges, just like we talked about with muscle and with nerve. And this results in a change in calcium flow. So this spike in the current is the fast block. That happens immediately. As a result of that two-polarization, we begin the slow block. And this is what you're going to be able to see in the sea urchin. Slightly different from the human, but similar structures occur. So just under the surface of the egg are these granules or vesicles that are called cortical granules because they're in the cortex. Okay? So as a result of that spike, and just the depolarization itself is the fast block, but as a result of that depolarization, these cortical granules will fuse with the plasma membrane surface of the egg and dump their contents into the space between the secondary oocyte and the zona pellucida. Okay? Now, I called it a secondary oocyte because it's just starting the process now of the second meiotic division. So it's a secondary oocyte with the sperms in chromosomes inside. It just hasn't divided yet. It will momentarily, but it's still a secondary oocyte. So these cortical granule changes do a couple of things. The zona pellucida separates from the egg. All right, you'll be able to see this if everything works out right in our sea urchins. You'll see this kind of swelling of the fertilization membrane, which is the equivalent of the zona pellucida, and hardens. And that hardening of the zona pellucida is the permanent nature structure that prevents sperm from entering into penetrating the egg. So, two blocks. The fast one is the sodium flow. The second one is cortical granules and calcium changes that allow the fluid to move out between the secondary oocyte and a hardening of the protein kind of cross-linking of the zona pellucida. So if we look at that over here with these models, so we have the ovulated oocyte. It's not showing the first polar body, but that would be present here. And this cell would be a secondary oocyte with chromosomal pattern of 2 parentheses in, meaning we have double copies of each type for 23 chromosomes. Out here would be the granulosa and follicular, rest of the follicular cells. This model shows the fertilization by the sperm with this little tail, photofilamentins that come off. And now we have the end chromosomes of the sperm actually inside the egg, but they haven't paired up with the secondary oocyte cells, and it still needs to undergo its second meiotic division, which it's going to do now. Again, there's still a first polar body there. So now we see the formation of the pronucleus, which is the male and female end chromosomes coming together to form a new cell that's 2 in. Still trapped under the zona pellucida, because when the sperm penetrates through it, doesn't just split it in half, it's only making a little hole. So still trapped under the zona pellucida are the first and second polar bodies. Okay. Now, as you can see, the cell starts to undergo division and forms 2 cells and 4 cells and so on, and all of that is still within the zona pellucida. And the cells keep getting smaller and smaller because they're still inside the zona pellucida. Each of those is an identical 2 in chromosomes. And the chromosomes are identical for one. This is just mitosis. So each cell is going to be identical in chromatic material to the one beside it. And this goes on until we have thousands of little cells. And it forms a solid ball of cells as it's moving through the oviduct. So here you can see the sperm coming in. And not only does that trigger the block the polyspermy, but it triggers the second meiotic division of our egg, our secondary oocyte. And the first polar body may or may not undergo division. Okay? So if it doesn't, if the first polar body doesn't undergo meiosis, then we would have our ootid that's in, and our first polar body that's 2 parentheses in, and our second polar body that's also in. Does that make sense? All right, so we have our secondary oocyte, first polar body. Each of these is 2 parentheses in. Fertilization occurs. We now have our ootid, which is in, the sperm over here, which is in, our first polar body, which is 2 in, and our second polar body, which is in. Okay? Then the same series of divisions and chromosomal changes occur in the male. So always go back to that sequence where it was easy to follow the X and the Y. And just remember that the amount of cytoplasm associated with each cell division is different. All right? So we have an uneven distribution, but what's inside each cell is exactly the same as we saw for the sperm. So this is called the pronucleus. So we have the male pronucleus and the female, each of which are in now. The pronuclei are the in chromosomes of each male and female cell. So up here, we have the ovum. It's the same thing as the ootid. We have the second polar body, and in this case, they've shown the first polar body having divided. We won't worry about that. And the 2 pronuclei, the 2 in chromosomes, move together. And then spindle fibers associate between them, right? And they start to undergo now mitosis, and already those new chromosomes in this new cell, and the newly arranged chromosomes, undergo replication and line up for their first cell division of mitosis. And so we see a 2-cell stage. So this is now a zygote, where we have the sperm in chromosomes associated with the egg in chromosomes to form a 2-in cell. So a zygote is now 2 in, not 2 parentheses in or in. It's now 2 in, a diploid cell, just like any other cell in your body that's not a gonadal cell. And so when it divides, it's going to undergo the same type of mitotic division. And it's going to undergo 2 and 4 cell stages, which were started in the sea urchins yesterday. All right, that can take a couple of days to occur. A morula is what we call a solid ball of cells. It still has the zona pellucida around it. It's a solid ball of cells. All of them are still 2 in. And then now, just as it's moving closer to the isthmus, that solid ball of cells develops a hollow center, which is a single layer of cells on the outside. Now, if we look at the solid ball of cells, we have different environments. The cells that are the very closest on the surface are going to have access to the highest concentration of hormones and chemicals in the oviduct. The cells that are deepest in that solid ball are going to be the farthest away. And that helps to determine what they do. All right? Then they hollow out. This is what is going to become the placenta. And the inner cell mass is, as we'll see tomorrow, is going to become our embryo and the amniotic membrane that surrounds the embryo. This is what it looks like as it enters into the uterine cavity, is as that blastula or blastocyst. Still has the zona pellucida around it, okay? So this is where we'll start tomorrow. Once it's within the uterine cavity, so it makes it through there usually in about 4 to 5 days, and then it takes another day or two to implant. So implantation is usually day 7 to 9, all right? Before it implants, the blastocyst loses its zona pellucida. That process is known as hatching. And that ruptures and releases the blastocyst. It would be very difficult for it to implant with that protein coat around it. All right, so now it's ready to settle into the uterine wall. So fertilization occurs within the ampulla, and then the zygote is formed. It undergoes all of its gradual changes, and then enters the uterine cavity as a blastula typically. All right, so let's stop there. We have a quiz. So before, while I'm setting up with the sea urchin fertilization, why don't you guys take 9:30, 11:20. Why don't we take the quiz at about half an hour? Okay? So sometime between 9:55 and 10. And that'll allow you a little bit of time to take a break and capture a last minute. So this is just on male reproductive. Okay?